close your eyes for just a moment and try to picture the most intricate, perfectly synchronized dance you can possibly imagine. Now think about that dance happening inside every single one of your body's cells, every second of every day. It's this breathtaking performance of growth, copying itself, and then dividing with incredible precision, constantly unfolding. This is, the fundamental biological process that really underpins all life, all growth, even how our bodies heal. Our mission today is to understand the secret choreography, if you will, that cells follow to pull this off. We'll explore the sophisticated molecular systems keeping it all in check, and, well, what happens when that rhythm gets lost. All the insights for this deep dive come from some really detailed medical education lecture notes giving us an expert-level view into this cellular ballet.
And it really is like a ballet, or, maybe more like a super precise, automated factory line. Understanding this whole process isn't just for biologists, it's truly foundational to medicine. It explains how we grow, how tissues repair themselves, and really crucially, how diseases like cancer kick off when these precise controls go wrong. It's the bedrock of what makes us, well, us.
So let's pull back the curtain on this rhythmic dance. The cell division cycle, often called the CDC, it's not just one chaotic thing, right? It's really structured with distinct stages. Each stage has its own job. Can you walk us through those stages? What are the cells actually doing at each step?
Absolutely. Think of it like a journey with clear milestones. Cells often start in a sort of resting phase, sometimes called quiescence. A good example is maybe a fully developed neuron. Yeah. Once it's formed, it usually just does its job, sends signals, doesn't divide anymore.
Okay, so GRO is like off the clock for division.
Kind of, yeah. But for cells that are going to divide, they enter G1, the GAP1 phase. This is really their growth and prep stage. They're busy synthesizing lots of proteins, specifically the ones they'll need for DNA replication. Then comes the crucial S phase. S is for synthesis. This is where the cell meticulously duplicates its entire genetic blueprint, all the DNA.
Every single bit.
Every single bit. After that, they move into G2, another GA phase. Here they make more proteins, but this time the ones needed for the actual division, for mitosis itself. And then the main event, M phase. Mitosis, that's the dramatic division of all the nuclear material.
Okay, so G1, S, G2, prep, copy, more prep, and then M, the division. And I know G1, S, and G2 are often sort of lumped together. Why is that?
Yeah, they're grouped as interphase. Basically, this whole period is when the cell is incredibly active metabolically. It's doing all the heavy lifting, getting everything ready for the huge event of mitosis. If you looked under a microscope during interphase, you wouldn't see those distinct X-shaped chromosomes. They're more like loose spaghetti in the nucleus.
Yeah.
But the cell is definitely busy. It's preparing for this incredibly precise split, making sure each new cell gets a perfect copy of absolutely everything.
It sounds like an astonishing feat of coordination.
Yeah.
What's one aspect of this preparation that you find particularly, mind-blowing?
For me, it's the sheer scale and the accuracy. Every single chromosome, every single piece of DNA has to be perfectly copied and then divided up exactly equally. This cycle is fundamental to, everything, from how you grew from a single egg to how your skin or your gut lining repairs itself constantly. Even tiny deviations, tiny missteps in this super tightly controlled sequence can have really significant, often devastating consequences. It's truly life's ultimate quality control system.
Okay, and speaking of quality control, if the cell cycle is this complex dance, there must be like a whole cast of characters, right? A molecular orchestra, making sure every step is perfect.
Yeah.
What are the key players conducting this show?
You're right, and this is where it gets really fascinating. The cell has this incredibly elegant molecular control system. We can think of maybe 6 main types of protein players. First, you've got cyclins and cyclin-dependent kinases, or CDKs. These two are like the main conductors.
Okay.
Then you have CDK inhibitors, CDKIs. They act as the brakes.
Right.
And then we also have Wee kinase, CDC25C phosphatase, and CDK activating kinase, or KEEK. A whole team.
Okay, wow. So cyclins and CDKs are the conductors. What exactly are they doing?
Well, the cyclins are kind of like the timekeepers. Their levels, their concentration in the cell, rise and fall really predictably throughout the cycle, almost like a schedule. CDKs, on the other hand, they're the workhorses. Their protein kinases, think of them as specialized enzymes that add a little tag, a phosphate group, onto other proteins.
On that switch.
Exactly like a switch. That tag can turn those other proteins on, turn them off, or change what they do. But here's the crucial part. CDKs are only active when they're bound to their specific cyclin partner, and only at the precise moment they're needed in the cycle. So you have different cyclin-CDK pairs for different jobs. G1S phase CDKs get things ready for DNA copying, S phase CDKs manage the copying, and mitotic CDKs drive the actual division.
Okay. So the cyclins basically tell the CDKs when it's time to act, and the CDK inhibitors, they're sort of the unsung heroes, making sure things don't just, run wild.
Precisely. CDKIs are absolutely crucial. They act like molecular brakes, putting a stop to CDK activity when it's not needed or if something's wrong. Proteins like P21, P27, INK4, they're really important examples. Then you have these other enzymes like kinase. It puts a specific stop tag, another phosphate group, on the CDKs, inhibiting them temporarily. Its opposite number is CDC25C phosphatase, which removes that same stop tag, activating the CDK. And CK is another activator, adding a different kind of go tag. It's this constant interplay, this push and pull of activation and inhibition.
That's a lot of molecular back and forth. Can you give us maybe an analogy? How do all these different molecules know when to act and when to hold back? It sounds like a finely tuned clock, but how does the cell make sure the tick-tock is just right, especially for that big decision to, copy itself?
Yeah, it's like hitting a critical threshold, a cellular point of no return. Let's look at the transition from G1 into S phase the moment the cell commits to replicating its DNA. It starts with signals from outside the cell, growth factors. These stimulate the activity of the G1 CDKs. Now, these active CDKs bound to their cyclin D partners start modifying a key protein called RB.
RB?
Think of RB as like a molecular clamp holding onto another protein called E2F. E2F is essential for turning on the genes needed for replication. When RB gets modified by the CDK, it lets go of E2F.
Actually releases the clamp.
Exactly. And here's the really clever bit. E2F, once it's free, actually triggers the production of more cyclin E, more CDK2, and even more E2F itself.
Wow, a positive feedback loop.
A really powerful positive feedback loop. It's like a chain reaction that rapidly ramps up, pushing the cell past that critical restriction point. Once it crosses that line, it's irreversibly committed. It will duplicate its DNA. There's no going back.
So once it hits that gas pedal, it's floored it all the way to S phase. Okay. And what about getting ready for the actual division, for M phase?
Right. Another critical transition happens at the boundary between G2 and M phase. Here, a different cyclin, cyclin B, starts accumulating during G2. It binds to its partner CDK, which is CDK1. Initially, the CDK1 cyclin B complex is held inactive, kept in check by that kinase we mentioned earlier.
The inhibitor.
Yep. But right at the moment the cell is truly ready to divide, kinase gets switched off, and CDC25C, the activator phosphatase, gets switched on. This removes the inhibitory tag from CDK1, activating the whole CDK1 cyclin B complex. This active complex is like a master switch for mitosis. It phosphorylates dozens and dozens of proteins, triggering all the dramatic events: chromosomes condensing, nuclear envelope breaking down, spindle forming. Everything.
Wow.
Then, at the very end of M phase, after the chromosomes have separated, cyclin B itself is rapidly targeted for destruction. This inactivates CDK1, allowing the cell to exit mitosis and finish dividing. It's this beautiful, intricate dance of activation and inhibition, phosphorylation and dephosphorylation, making sure every single step happens in perfect order.
With all that precision, there must be some really serious quality control happening, right? Which I guess leads us to the guardians of integrity.
Yeah.
The checkpoints and the DNA damage response. The cell have these built-in surveillance systems to make sure everything's okay, especially the DNA and the chromosome distribution.
That's absolutely right. If DNA gets damaged, the DNA damage response system kicks into high gear. You can imagine it like a team of highly specialized sensors. Proteins like ATM and ATR are key players here. They recognize different kinds of DNA damage. Once damage is spotted, these sensors trigger a signaling cascade, activating other kinases downstream like CHASE-K1 and CHK2. These act like stop signs. They halt the cell cycle.
So they literally put the brakes on. And they do this at specific points, like gates slamming shut.
Exactly. For instance, if DNA damage is detected in G1, before replication even starts, they shut down a key enzyme needed to initiate replication, so the cell won't even begin copying damaged DNA.
Smart.
Very smart. And later, say in S phase while DNA is being copied, or in G2 after it's copied, if damage is found, the signals prevent the activation of those mitotic CDKs we just talked about. This essentially arrests the cell in G2, stopping it from entering mitosis until the damage can hopefully be repaired. The cell absolutely will not charge into mitosis with damaged DNA if these systems are working.
And then there's P53. I've heard it called the guardian of the genome.
Sure.
What makes P53 so special, so powerful?
It's a very fitting name for it. When DNA damage occurs, P53, which is a type of protein called a transcription factor, gets activated. Its main job is to act as a tumor suppressor. One of the key things it does is dramatically ramp up the production of a protein called P21. Remember P21? It's one of those CDK inhibitors.
That breaks.
Exactly. P21 puts a universal brake on all the cyclin-CDK complexes, effectively causing the cell to arrest, to stop, in both G1 and G2. This buys the cell precious time to try and repair the DNA damage. But P53 has another, even more dramatic function. If the DNA damage is just too severe, too extensive to be repaired properly, P53 can switch gears and activate genes that trigger apoptosis.
Programmed cell death.
Programmed cell death, exactly. Cellular self-destruction. This is crucial because it ensures that severely damaged cells, the ones that might become cancerous, are eliminated before they can cause harm. This dual-role cell cycle arrest for repair, or apoptosis if repair fails, is why P53 is so incredibly important. And it's why nearly all human cancers show mutations in either P53 itself or in the pathways that control it. Losing P53 is a major step towards uncontrolled growth.
Wow. Okay. And beyond just the DNA code itself, cells also have to make sure they divide the chromosomes correctly, right? That each daughter cell gets the right number. That's where the spindle assembly checkpoint, or SAC, comes in. That sounds like another big job.
It's absolutely vital. The SAC pathway is designed specifically to prevent chromosome segregation, the actual pulling apart of the duplicated chromosomes during anaphase, until every single chromosome is perfectly attached to the mitotic spindle fibers. Imagine it like a security system at the chromosome level. If even one chromosome isn't properly hooked up to the spindle, specifically at its kinetochore, it sends out an unattached signal.
A warning signal.
A biochemical warning signal, yes. This signal activates a complex of checkpoint proteins. These proteins then act like a molecular brake, specifically inhibiting the machinery, a complex called APCC that's needed to initiate anaphase, the chromosome separation step.
So it's basically yelling, hold on, not everyone's ready yet.
Pretty much. It puts the brakes on division until every single chromosome confirms it's properly attached and aligned correctly at the cell's equator. It's a real perfectionist.
Okay.
Once all chromosomes are attached correctly under the right tension, that unattached signal stops being sent. The molecular brake, the checkpoint complex, is then released or disassembled. This allows the APCC machinery to become active, degrade some crucial proteins holding the chromosomes together, and finally trigger anaphase. The chromosomes can then safely separate. It's an incredible system ensuring genomic integrity.
So what does this all really mean for us? I mean, these checkpoints, they sound like these incredibly meticulous quality control stations. They're making absolutely sure everything is perfect before the cell dares to move forward. What are the profound consequences if these critical systems fail?
The consequences are, frankly, severe. These surveillance mechanisms, the DNA damage response, the spindle checkpoint, they are the absolute bedrock of maintaining genomic stability from one cell generation to the next. Losing them, having them malfunction, is a primary driver of disease and especially cancer. Without these vigilant guardians, cells carrying damaged DNA or cells with the wrong number of chromosomes can just continue to divide unchecked. They accumulate more and more errors, potentially leading directly to serious pathology. It's a dangerous, slippery slope.
Right. Which brings us to, I guess, the darker side of this cellular dance when it goes wrong, specifically in cancer. How can systems that sound so precise become so dangerously out of control? It almost feels like the cellular dance just loses its rhythm entirely and goes completely wild.
It truly does. Cancer, at its core, is cell cycle dysregulation. It's that cellular dance, completely offbeat, out of control. This usually happens through two main kinds of genetic changes. First, you can have what are called loss-of-function mutations. This is where a gene that normally acts to prevent cancer, a tumor suppressor gene like P53 that we just discussed, or RRET's inactivated, it breaks. It's like the cellular brakes just fail completely.
Okay, so the cell loses its ability to slow down or stop when it should.
Exactly. The other main type is a gain-of-function mutation. Here, a different type of gene, one that normally promotes cell growth in a controlled way, acquires increased activity or even a new abnormal function. It becomes what we call an oncogene. Think of genes like RAISE, which signals growth, or even cyclin-D, one of the cell cycle regulators. They become like stuck accelerators, constantly pushing the cell to divide, even when it shouldn't. So cancer often results from this dangerous combination, the brakes failing and the accelerator being jammed down.
And what are the sort of downstream effects, the molecular consequences of those kinds of changes?
We see several common patterns of dysregulation in cancer cells. For instance, you often see the overexpression of cyclins and CDKs, too many conductors, too many workhorses driving excessive cell division, pushing the cell through the cycle way too fast. Obviously, the loss of those key tumor suppressor genes like P53 and ARV is huge. They normally halt the cycle for repair or trigger apoptosis, so losing them lets abnormal cells just keep proliferating without consequence.
Right.
Similarly, you might see reduced levels or inactivity of the cell cycle inhibitors, those CDKIs like P21 and P27. That removes crucial breaking power, making the cell much more permissive to just keep dividing. And this whole dysregulation process itself can lead to profound genomic instability. The cell starts making mistakes, copying its DNA, chromosomes break or get rearranged. And if the DNA repair systems are also faulty, which they often are in cancer, this just accelerates the accumulation of genetic chaos.
It really sounds like a spiral into, well, cellular anarchy.
It really is. Clinically, we often see cancer cells showing elevated markers of proliferation. We can measure things like Ki-67 levels, which indicate actively dividing cells. And critically, the dysfunction or even complete loss of those vital cell cycle checkpoints, we talked about the G1S checkpoint, the G2M checkpoint, the spindle checkpoint, allows these deeply flawed cells to continue dividing, directly fueling cancer development and progression. So yeah, these molecular changes, these failures in the cell cycle control system directly create the defining characteristics, the hallmarks of a cancerous cell: uncontrolled growth, resistance to normal stop signals, and the ability to evade those crucial quality checks.
Okay, let's shift focus a bit now and zoom in on the grand finale itself, mitosis in action. What's actually happening, on the ground level as the cell divides? You said it's more than just splitting in two. It's like an incredibly precise reconstruction.
It absolutely is. Mitosis technically is the division of the nucleus. It's usually followed immediately by cytokinesis, which is the division of the rest of the cell, the cytoplasm. The whole goal is to produce two genetically identical daughter cells, each getting a perfect, complete set of the duplicated chromosomes. It's a highly visual process, very dynamic, and we break it down into distinct phases. It starts with prophase. Here, the chromatin, which is usually loose in the nucleus during interphase, starts to condense. It coils up tightly into those distinct, visible chromosomes that you often see in diagrams, the X shapes. Also, during prophase, the nucleolus, a structure inside the nucleus, disappears, and the cell starts building the mitotic spindle, that crucial machinery for separating the chromosomes.
So the chromosomes are getting packed up, organized, like they're preparing for a big move.
Exactly like that. Then comes chromatophase. This is where the nuclear membrane, the envelope surrounding the DNA, completely breaks down and dissolves. At the same time, special proteins assemble onto the centromere region of each duplicated chromosome, forming structures called kinetotors. You can think of these like molecular handles.
Handles for the spindle to grab onto.
Precisely. Microtubules, the fibers growing out from the poles of the forming mitotic spindle, then reach out and attach to these kinetochore handles. Once attached, the chromosomes start getting pulled and pushed around. This leads into metaphase. Here, the spindle fibers work together to precisely align all the duplicated chromosomes right along the very middle of the cell. This imaginary line is called the metaphase plate. This perfect alignment is absolutely critical. It ensures that when the chromosomes split, each new nucleus will receive exactly one copy of each chromosome. No more, no less.
Like a perfect cellular lineup, everyone exactly in their assigned spot.
A perfect lineup, yes. That sets the stage for anaphase. This is the separation phase. The connections holding the two identical halves of each duplicated chromosome, the sister chromatids, braid apart at the kinetochore. Then the separated sister chromatids, now considered individual chromosomes, are pulled forcefully towards opposite poles of the cell. This movement is driven by the kinetochore microtubules shortening, literally reeling them in, and also by other spindle microtubules pushing the poles further apart. Finally, we reach telophase. The chromosomes arrive at their respective poles. They start to decondense, becoming less tightly packed again. And importantly, a new nuclear envelope begins to reform around each complete set of chromosomes, creating two distinct new nuclei within the single elongated cell.
And all this complex movement and organization is orchestrated by that mitotic spindle. That really sounds like the absolute backbone of the whole operation.
It absolutely is. It's the backbone, it's the scaffolding, it's the transport system, all rolled into one. The mitotic spindle is this incredibly dynamic, highly organized structure made of microtubules, the centrosomes that organize them, and many associated proteins. Its roles are critical: accurately segregating the chromosomes, ensuring that precise alignment at metaphase, enforcing that spindle assembly checkpoint we talked about earlier, and ultimately helping to position the plane for cytokinesis. It has to distribute that precious genetic material flawlessly to the two daughter cells.
Okay, and then that final step, cytokinesis, where the cell actually divides itself in two. How does it manage to pinch off and split everything perfectly down the middle.
Right. Cytokinesis is the final act. This is where the cytoplasm, including all the organelles like mitochondria and ribosomes, gets physically divided between the two forming daughter cells. In animal cells, this is driven primarily by something called the contractile ring. It's this powerful band made of actin filaments and myosin motor proteins, the same kinds of proteins involved in muscle contraction.
Like a drawstring.
Exactly like a purse string or a drawstring. It forms around the middle of the elongated cell, underneath the plasma membrane, and it progressively tightens, constricting and pinching the cell into two separate daughters. This process has to be tightly coordinated, both in space right in the middle, between the two newly formed nuclei, and in time, usually starting during late anaphase or telophase. A small signaling protein called RhoOA plays a key role here, stimulating the assembly of the actin filaments and promoting the contractile action of the myosin motors in the ring.
So the cell basically built itself a temporary muscle to pinch itself in two. And what about the nuclear envelope during all this? You mentioned it dissolves in prometaphase. How does it magically reappear in telophase?
Yeah, that's another incredible piece of this cellular precision engineering. The nuclear envelope, which normally protects the DNA, has to completely disassemble to allow the spindle fibers access to the chromosomes. This disassembly is triggered by those active mitotic CDKs, CDK1 cyclin B. They phosphorylate, add phosphate tags to the lamin proteins. Lamins form a meshwork lining the inner nuclear membrane, providing structural support. Phosphorylating them causes this lamina meshwork to break down, leading to the fragmentation of the nuclear envelope.
Okay, phosphorylation breaks it down.
Right. Then, at the very end of mitosis, during telophase, as CDK1 activity plummets because cyclin B is destroyed, other enzymes called phosphatases swing into action, specifically PQ1 and PP2A. They remove those phosphate tags from the lamins. This dephosphorylation is the trigger. It allows the lamin proteins to reassemble their meshwork on the surface of the segregated chromosome masses. This, in turn, recruits fragments of the old nuclear membrane, which have been dispersed in the ER, to come back together and fuse, reforming two complete nuclear envelopes.
Wow. And what about the spindle? It's still there in the middle, isn't it?
It is, momentarily. There's actually another fascinating step. Special protein complexes, called EESCRT machinery, are recruited. First, they recruit another protein, spastin, which acts like molecular scissors to sever the remaining spindle microtubules connecting the two nascent daughter cells. Then, the ESCRT proteins themselves help promote the final sealing of the reforming nuclear envelopes. It's just an incredible feat of cellular construction and deconstruction.
The sheer level of coordination involved in mitosis and cytokinesis is just astounding. It really highlights how absolutely vital this precision is for preventing genetic errors in the new daughter cells.
Absolutely. The complexity of the cellular machinery, the choreography, the timing, it's truly mind-boggling when you think about it happening correctly billions of times a day in our bodies. Any significant misstep here, like missegregating even one chromosome, can lead to daughter cells with an abnormal number of chromosomes aneuploidy, which has severe consequences for their viability and function and is a powerful driver of diseases like cancer and developmental disorders. It's a testament to the fundamental robustness of life, but maybe also highlights its inherent fragility if these systems fail.
That's a really powerful thought, and it raises an important question. How does this incredibly complex, sometimes fragile cellular machinery manifest so profoundly in, say, a child's health? Let's maybe turn to a real-world example now, ataxia telangiectasia.
Yes, let's consider a clinical case. Imagine a 20-month-old boy brought to a pediatric neurologist. His parents are worried because he hasn't really developed a steady walk yet. He's quite unsteady. Plus, he's had a history of recurrent ear infections, sinus infections, and even needed hospitalization once for pneumonia. Looking at him, he's perhaps a bit small for his age, but his intelligence seems normal. However, on examination, the neurologist notices abnormal eye movements, his eyes sort of dart around, they don't track smoothly, and he struggles to hold his gaze fixed on something. Some lab tests are run. Key findings include a high level of serum alpha-fetoprotein, which should normally be low after early infancy, and a notably low level of serum IgA, an important type of antibody for fighting infections.
Okay, that's definitely a collection of concerning signs for such a young child. How does this connect back to our discussion on the cell cycle?
Indeed. This clinical picture is highly suggestive of a condition called ataxia telangiectasia, or AT for short. The genetic basis for AT is a defect, a mutation, in the AT gene, which is more formally known as the ATM gene, located on chromosome 11. This ATM gene encodes a really crucial protein. ATM is a large protein kinase, and it's a central player, a conductor, if you will, in the cell's DNA damage response system, those checkpoints we were just discussing. Fundamentally, AT represents a critical defect in the signaling system that normally slows down or stops the cell cycle when DNA damage is detected.
Ah, so those DNA damage checkpoints, the vigilant guardians we talked about,
Well,
in this child, they're simply not working properly because the ATM protein is faulty.
precisely. A lack of functional ATM protein means the child's cells lose a critical ability. They can't properly halt cell division at multiple checkpoints, like the G1S or intra-S checkpoints, when their DNA gets damaged, which happens spontaneously all the time, or from environmental exposures. They also can't fully activate all the necessary DNA repair pathways efficiently. And furthermore, they struggle to trigger apoptosis, that programmed cell death, even when faced with significant DNA damage that should trigger elimination. The damaged cells just keep going, trying to divide even when they shouldn't.
So because the cells can't reliably stop, can't fully repair damage, and can't properly eliminate the severely damaged cells, what does that ultimately mean for this child's health, for their life?
Tragically, the consequences are severe and progressive. Children affected with classical AT typically experience progressive neurodegeneration, particularly affecting the cerebellum, which leads to the ataxia, the problems with coordination and balance. They suffer from recurrent infections because the ATM defect also impacts immune system development and function, leading to immunodeficiencies like the low IgA we saw. And critically, they face a dramatically increased risk of developing cancer, particularly lymphomas and leukemias. This heightened cancer risk is a direct consequence of their cells' inability to cope with DNA damage. They are hypersensitive to radiation, for example, and the resulting profound genetic instability that accumulates because those critical cell cycle checkpoints and DNA damage responses are broken. This condition really powerfully underscores just how absolutely critical these functional cell cycle checkpoints and DNA damage response systems are, not just for maintaining the integrity of a single cell's genome, but for the overall health, development, and disease prevention of the entire organism of the person. It's a very stark reminder of the profound impact this intricate cellular dance has on our actual lives.
Wow. We've really journeyed deep into this incredibly intricate world today, haven't we? From the cell cycle's rhythmic stages and that elaborate orchestra of molecular regulators controlling it all, to the vigilant checkpoints standing guard against errors, the sheer wonder of mitosis unfolding, and then, the profound and sometimes heartbreaking consequences when these intricate systems fail, whether that leads to the uncontrolled proliferation of cancer or the devastating multi-system impacts of a condition like ataxia-telangiectasia. What really stands out for me is just how incredibly intricate, how essential, and yet how fundamentally vulnerable this process is. Every moment, billions of cells inside you are executing this complex dance with, most of the time, breathtaking precision, often completely without you even realizing it.
Absolutely. And maybe that leads to a final thought for everyone listening. Given the extraordinary precision and complexity required for every single cell division to happen correctly, what does this deep dive into the cell cycle, its regulators, its checkpoints, what does it tell us about the fundamental robustness or perhaps even the surprising fragility of life itself? And taking that a step further, what implications might our growing understanding of this incredible cellular choreography have for the future, for developing new medical breakthroughs against diseases like cancer, or maybe even for understanding the fundamental processes of aging, and perhaps one day how we might extend not just lifespan, but healthy lifespan? Definitely something to ponder.